Hi, so we're going to review mitosis. So first off, you can watch this cell division by Hank Green. I'm not going to show you that on here. Yesterday we talked about this a little bit. Cell size limitations. What are some reasons that cells divide? Because the DNA overload. The cell becomes too big and there's too much for the DNA to do. There's too much to control. If you think about like a really big town like Eau Claire, they probably have a principal and an assistant principal. They have two just for the high school. Whereas our school is smaller and we only have one principal, 6 through 12. So if you think of the principal like the DNA, helping to control everything of the school, if the school is a cell, you need to have two in order to make it work. So our school only has one because it's small yet. So when this cell gets too big, it needs to divide because it's too much for the DNA to control. And then we talked about the surface area is too small. You need a very large surface area to volume ratio. So if you look here, this little tiny baby cube has a small surface area and a small volume, but the ratio is really big. 6:1 would be the ratio. And this one here, the ratio is 1.2:1 because although the surface area is a lot bigger, so is the volume. And so the surface area and volume are very similar in size, so the ratio is like 1.2:1. So it can't efficiently get rid of all the waste and take in enough nutrients to make it to keep up with everything. Think of it like your one-bedroom apartment. You can run in and run out and get all your things really quickly and whatever. Whereas if you forget your cell phone or your water bottle or whatever your favorite thing is in your bedroom of your mansion with 12 bedrooms, it's going to take you a lot longer to go get it. And that's the same way with the cell. Okay, so chromosomes is DNA that's condensed. So they're a thread-like structure within the nucleus. They contain the genetic information, which we know passed on from one generation to the next. And during mitosis, they become visible. So chromatin kind of is made up of DNA coiled around histones, which are proteins. And when that chromatin condenses, it makes the chromosomes. Two sister chromatids right here make up a chromosome. Centromere is the part in the middle where they meet up. So you can see this. Here is a chromatid. Here is a chromatid. Here's the centromere in the middle where they connect. This whole thing is a chromosome. So this is kind of like a bowl of spaghetti, and that's chromatin. That's what your nucleus looks like when you look at it under interphase. And then during prophase, these chromosomes become visible because all this spaghetti helps to coils up into the chromosomes that make up your genetic material. Okay, so what do cells have to do to prepare for cell division? Well, what do you have to do to prepare for anything? If you're going on a trip an hour away, you probably will fill up a water bottle, or you'll grab some money if you want to buy your own water and waste plastic. You'll probably go to the bathroom because it's an hour away, right? You might pack a snack, or you'll pack money to buy food. Maybe you have your phone and want to bring your phone, or you worry about your battery dying. You prepare, right? You prepare for you to go on this trip, hour away, 4 hours away, whatever. Cells have to prepare for cell division as well. So how do they do that? Well, look at this blue writing here. That's interphase. Your cell spends most of its time in interphase. That's interphase is G1, S, and G2. G1 is where the cell grows. See, look at little tiny cell gets bigger. S phase is the synthesis phase. That is where the cell is duplicating its DNA. Synthesis in biology means to make. So it's making a second copy of DNA. And we're talking about mitosis, so it's making identical copy. Because remember, mitosis is the division of body cells or somatic cells. So that would be like your earlobes, your muscles, your stomach, your skin, things like that. They're all body cells. So the two that are produced during mitosis are identical to the other ones. G1, S, synthesis, makes a double set of DNA. G2, then that's where it prepares. That's where it's like, okay, I'm going to double my mitochondria. I'm going to double my Golgi apparatus. I'm going to prepare for mitosis. I'm going to get everything ready. So that's how that works. So here it kind of shows you interphase, what's all happening. A homologous pair of chromosomes have two sister chromatids. So two sister chromatids make up one homologous pair of chromosomes. Prophase is the first stage, the chromosomes condense. Metaphase is the second stage, chromosomes line up in the middle. Anaphase is the third stage, the chromatin Chromatids, so it splits at the centromere and then goes to opposite parts of the cell. Telophase is the fourth stage and it kind of goes back to normal. Now you have itty bitty cells. And cytokinesis is the last stage, and that's where they divide into cell plasm. Cytoplasm divides into two. So that is mitosis in a nutshell.